Hi everyone, this is Dr. LeBlanc, and this is part 1 of 3-part lecture on Chapter 12, the organization and packaging of DNA. Part 1 deals with the prokaryotic genome, its organization and the action of a very special enzyme called DNA+Gyrase. Parts 2 and 3 will talk more about eukaryotic DNA. Here's a bacteria cell, an E. coli cell, shown in this electron micrograph, with all of its DNA contents spilled out into the surrounding media. Now, all of this DNA has to be packaged tightly and in a highly organized fashion inside the bacteria cell so that it can be accessed for very important functions like DNA replication and transcription into messenger RNA and then subsequent translation into protein. Genomes exist in all organisms. Prokaryotic organisms typically have one single chromosome that contains all of the genetic material that makes up a prokaryote. Prokaryotes are bacteria and viruses. They have no nuclei and are typically much smaller than eukaryotic organisms. Eukaryotic organisms have a nucleus into which all of the DNA that comprise the genome is put. They also have mitochondrial genomes and chloroplast genomes if you are a plant. Now, chromosomes are the small packets of DNA that make up the genome. As I said before, prokaryotes typically have a single chromosome, whereas eukaryotes have multiple chromosomes. Their genomes are divided into multiple chromosomes in the nucleus. However, mitochondria and chloroplasts are more like prokaryotes, and we'll talk about that at a future point. Mitochondria and chloroplasts are more like prokaryotes and have a single chromosome. Here's a typical prokaryotic genome that is a single chromosome, that is a circular chromosome. Okay? Eukaryotes do not have circular chromosomes, we have linear chromosomes. This DNA in this single chromosome can be annotated. We can find regions that encode genes, here indicated by these black lines. We can find regions that contain repetitive DNA sequences, in other words, DNA that's the sequence of the nucleotides is repeated over and over and over again. And these are probably relics of past viral infections. Now, we also have some intergenic regions, DNA, which we don't know what the sequence, the function of the sequence is. It may have a function, but we don't know what it is just yet. There is also a very important region in all chromosomes, circular or linear, called the origin of replication. The origin of replication is a sequence of DNA nucleotides to which the machinery that's going to replicate this entire genome can bind. Okay, it's a very specific sequence of nucleotides, and there's a very specific and complex set of proteins that binds here to initiate DNA replication. Once the chromosomal replication machinery has bound, DNA replication can proceed in two directions, usually in a prokaryotic genome. Here's a bacteria cell that has been stained with a fluorescent dye that will detect DNA. So here's a region of DNA that contains DNA here, and this cell that is dividing. This region inside the bacteria cell where you find the prokaryotic genome is called the nucleoid. It is not a nucleus. It is not membrane bound. However, it is a region where the DNA is concentrated, and it is actually bound to the bacterial cell wall membrane. Now, we have to get all this DNA packaged into the bacteria cell. To fit inside the cell, it has to be compacted about 1,000-fold. There are proteins that bind to specific DNA sequences all along the circular chromosomal molecule, and they organize the DNA into loops. These proteins bind to the DNA, and then just like a purse string, they bind to each other and create these loops that makes the DNA, the chromosome, look like a flower here. The loop structure compacts the chromosome approximately 10-fold, but we need to get it even more compact than that. The number of loops varies according to the size of the bacterial chromosome and the species. For E. coli, a typical loop has approximately 40 to 80,000 base pairs, and the entire chromosome is organized into about 50 to 100 loops. So you have 50 to 100 petals on the flower, and each of the petals is approximately 40,000 to 80,000 base pairs of DNA. Now, in order to get this compact even more, these individual loops become supercoiled. DNA supercoiling is a second very important way to compact the bacterial chromosome. How is this accomplished? Supercoiling within loops creates a more compact chromosome. Well, let's look a little bit at some of the math that's involved in supercoiling. A typical B structure DNA has approximately 10 base pairs per turn. That's the normal thermodynamically stable form of a DNA, double-stranded DNA molecule. If you turn it and underwind it so that instead of 10 base pairs per turn, you have 12.5 base per turn, this creates an unstable structure. And so what happens is some of the DNA gets looped out so that the linear portion returns to its 10 base per turn, and you have what's called a single negative supercoil. Okay. Alternatively, if you overwind it, you put more coils in than should be there in a thermodynamically stable, and you have a fewer number of base pairs per turn because the coils are closer together. You have this results in a positive supercoil, where the linear portion again is in its thermodynamically stable 10 base pairs per turn, and you have another bulge here, another positive supercoil. So DNA supercoiling can influence the chromosome structure. Okay. In E. coli, there is one negative supercoil per 40 turns of the double helix, typically. Negative supercoiling helps the compaction of the chromosome, but it also creates tension that may be released by DNA strand separation. Okay? If you have a region of base pairs here that is less stable than its surrounding DNA sequence, then these strands will separate if there's a negative supercoil. Think about the structure of the bases. A base pair, an AT base pair versus a GC base pair. How could that sequence of nucleotides in this negative supercoiled area influence the tendency of the strands to separate? I want you to think about that. This enhances DNA replication and transcription, having these negative supercoils. The enzyme that accomplishes that typically in chromatin is called DNA+Gyrase. If you have a circular chromosome and you add DNA+Gyrase and a couple of molecules of ATP, you can create negative supercoils. And I'm going to go through this diagram here. Okay, here's our typical gyrase. It has 4 subunits, 2 red and 2 green here, the A and the B subunits. And DNA binds into this groove here in the lower jaws. The A subunit and then loops around and comes back out through, just like this. DNA gyrase then cuts the DNA on the bottom strand here. And the top strand is forced through while the DNA gyrase holds on to the ends here. It's cut apart. Now you have DNA in a completely different configuration where this bottom strand is the strand that started out on the bottom ends up on the top as the DNA gets looped around this DNA gyrase. The ability of gyrase supercoils into DNA is crucial, absolutely crucial, for the bacteria to survive. If the DNA gyrase is inhibited, the bacteria cell will not survive. So blocking the function of this enzyme, blocking the function of DNA gyrase, is a way to cure or alleviate bacterial diseases. Two main classes of drugs inhibit DNA bacterial topoisomerases. This is the topoisomerase is the big class of enzymes to which DNA gyrase belongs. Those two classes of drugs are quinolones and coumarins. And one important feature for our benefit is that these drugs, the quinolones and the coumarins, do not inhibit eukaryotic topoisomerases because, as you might see, eukaryotes also have topoisomerases to introduce. And these drugs do not inhibit the eukaryotic ones, but they do inhibit the bacterial DNA gyrases. One example is ciprofloxacin, or Cipro, which you may have heard of before. It's used in the treatment of anthrax and other diseases. Here's a couple of molecular diagrams of some coumarins, novobiacin and chlorobiacin. They inhibit the hydrolysis of ATP by binding at a site in the enzyme that overlaps the ATP binding site, acting as competitive inhibitors. There's a couple of animations for you also on Blackboard that you can watch that describe compaction and the action of DNA gyrase.